Okay, so welcome back to this next video in which we are discussing the GQ pathway and the palmitoylation of IP3 receptors. Okay, right. So, we are going to be interested in the downstream signaling pathway of this IP3 molecule. So we've discussed that when you activate the GQ pathway, the two overall effects that this has is that it produces diacylglycerol and also inositol 1,4,5-trisphosphate. Now, I've said diacylglycerol is absolutely fascinating, but we're not going to discuss it here. We're going to focus on the inositol 1,4,5-trisphosphate, the IP3. Okay, now IP3 is going to go into the cytoplasm of the cell, and it's going to diffuse over to the intracellular organelle that is the endoplasmic reticulum. Okay, and in the endoplasmic reticulum, you have a large store of calcium. Okay, so let me show this. So the endoplasmic reticulum, again, you have a phospholipid bilayer here. Okay, and in the endoplasmic reticulum you have very high calcium levels, much higher than you have in the cytoplasm. Indeed, calcium level in the cytoplasm of the cell, so let's say this is the cytoplasm of the cell outside of the endoplasmic reticulum, calcium level in the cytoplasm is around 100 nanomolar. Okay, so that is very, very low. Okay, if you compare that to potassium concentration within the cytoplasm, which is around 155 millimolar, okay, so over 150, over a million times bigger, 1.5 million times bigger, basically, is the potassium concentration, and even compared to the sodium concentration, which is around 12 millimolar, and generally sodium within cells is thought to be pretty low. You know, that's massive compared to this. Huge, absolutely huge, you know, good 100,000 times bigger. So calcium concentration within the cytoplasm is extremely low. Okay, now, you do, however, have a high concentration of calcium in this intracellular organelle, which is called the endoplasmic reticulum. And the endoplasmic reticulum is often abbreviated to ER for short. Okay, now, basically, there is a way of releasing the calcium from the intracellular store temporarily into the cytoplasm, and this calcium can then have all sorts of signaling activity, basically. And the way that IP3 is going to cause the release of calcium from the ER stores is that it's going to work on a protein or a receptor that's within the plasma membrane, or, well, sorry, the membrane, the phospholipid bilayer of the ER, so is within the ER membrane. Okay, and this receptor is known as the inositol 1,4,5-trisphosphate, or IP3 receptor, so R for receptor. Okay, now let's have a look at the structure of this receptor in a bit more detail. So I'll pull it out here and we'll draw a big picture of it. Okay, so here now is a big picture of this IP3 receptor. Okay, so fundamentally it is a calcium channel. It's going to allow calcium to move through it into the cytoplasm. Okay, and remember, this is the membrane of the ER, so this side is the cytoplasm, and this side is the lumen of the ER. So don't be confused into thinking that this is the extracellular fluid and this is the intracellular fluid. This is the ER membrane, and therefore we're dealing with the cytoplasm and the lumen of the ER. Okay? And it's a channel, so it fundamentally has a pore going through it, which will allow calcium ions to move through. Now, it's not just made up of one polypeptide. Instead, it's made up of a bunch of polypeptides stuck together, and specifically it's made up of 4 separate polypeptides. So it is a tetramer, basically. Now, if I take one of these subunits of the IP3 receptor out, let's now have a look at the structure of that polypeptide, i.e. let's have a look at its membrane-spanning topology, basically, how many times it spans the membrane. Okay, right, so you have to remember when looking at this that this is the cytoplasmic side and this is the luminal side. So effectively the lumen is almost equivalent to the extracellular fluid. Basically, even though of course it's intracellular, the cytoplasm is one continuous lump of fluid, and this is effectively, it's analogous to the extracellular fluid with regards to this protein. It's going to allow things to move from here into the cytoplasm, just like you'd allow things to move from the extracellular fluid into the cytoplasm. Okay, so it kind of looks like an upside down equivalent of what you'd see in the plasma membrane for calcium channels, basically, or potassium channels, even better. But of course it is a calcium channel rather than a potassium channel. Okay, so basically you start off with the amino group over here. Okay, then it spans the membrane. So the amino group is initially on the cytoplasmic side. Then you have a membrane spanning alpha helix which spans the membrane. Then it curves around, spans the membrane again. That's the second membrane spanning alpha helix. Then again, third membrane spanning alpha helix. Fourth, fifth, then it has a little dip where it goes into the membrane and then doesn't quite make it, and then has the sixth membrane spanning domain before ending in the carboxylic acid group. Now, if you know anything about potassium channels, specifically 6TM P-loop potassium channels, this will look familiar. It's exactly what we would draw for a potassium channel, except that it's upside down, basically. And that's really because we've now got the cytoplasm on the top and the lumen, or what would have been, what is equivalent to the extracellular fluid, on the bottom. So that's why it appears upside down. Usually you'd have the cytoplasm on the bottom and then the extracellular fluid on the top if we were dealing with the plasma membrane rather than the ER membrane. So it's got a very similar structure to the subunits of potassium channels, basically, or at least 6TM potassium channels. Of course, there are other forms of potassium channels which it doesn't look like at all. Okay, right. So this is the membrane spanning topology of each one of these subunits. You build 4 of these proteins, and then you can stick them all together to make a tetramer like this. Now, the really simple thing would be if in the human genome you had one gene which coded for the IP3 receptor subunit, and all you had to do was transcribe this gene 4 times, get 4 pieces of mRNA, translate these 4 pieces of mRNA and stick them together to make an IP3 receptor. That would be the very simple scenario, i.e. that there would be one gene coding for this IP3 receptor subunit. Okay, the reality is that there are 3 separate genes which all code for slightly different IP3 receptor subunits. Okay, so there is the IP3 receptor gene 1, okay, called the IP3 receptor 1 protein. There is the IP3 receptor 2 protein, IP3R2, and there is the IP3 receptor protein 3. Okay, so there are 3 separate proteins. Now, they all have the same membrane spanning topology, okay, so that's nice and straightforward, but they will have slightly different sequences of amino acids, so they will differ slightly, basically. But they can all associate to form tetramers and make a complete IP3 receptor. So remember, these are genes coding for subunits of the IP3 receptor, not for the entire thing. The whole thing is made up of 4 of these stuck together. Okay, so this raises an interesting question. Can they form homotetramers? And do they form heterotetramers? And let me explain what I mean by that. The simple scenario would be then if, you know, to make an IP3 receptor you use the same subunit in all 4 slots. Okay, so for instance, if you wanted to make an IP3 receptor that contained IP3 receptor 1s, then what you'd do is you put 4 of them in each one of these slots, and your IP3 receptor, you could then call it an IP3 receptor type 1, if you like. Then you could do the same for 2 and the same for 3. That sort of a construction would be called a homotetramer, because you have used the same subunit in all 4 slots. Okay, now the reality is nature is more complicated than that. It can mix and match. It can use different receptor subunits in different slots, basically. So it could potentially use an IP3 receptor 1 subunit here, then an IP3 receptor 2 subunit here, maybe an IP3 receptor 3 subunit here. Basically, it's more complicated. It makes heterotetramers, okay, not just homotetramers. Okay, right. So, basically, IP3 receptors, therefore, they can be homotetramers, they can be heterotetramers, but they are constructed out of these 3 different subunit types. Okay, right. One more cool fact about the IP3 receptors is that each one of these subunits, each one of these polypeptides, is approximately 2,750 amino acid, and alpha is just shorthand for amino acid. So they're 2,750 amino acids long. That is a massive great protein. And then they're assembling into tetramers, so the whole receptor is going to be absolutely gigantic, basically. And these things really, really are. They are visible on electron microscopes, so they are massive, massive great things. Right, okay, so let's now discuss how IP3 is going to open these IP3 receptors. Okay, so this is where there is a lot of misbelief, basically. Most people would say that the IP3 binds to the IP3 receptor and that it actually causes the IP3 receptor to open. Now, that's a simplification. It's a good simplification, but it's a lie, basically. It's a helpful simplification. Very often it's a very helpful simplification, but it is a slight lie. So let me explain to you now what IP3 actually does. Okay, so let's now look at our IP3 receptor from the top here. So we're looking as though we're sitting in the cytoplasm looking at the IP3 receptor from above. Okay, so it's made up of these 4 separate subunits all joined together. Now, basically, initially, before IP3 has gone up in the cytoplasm and before it's bound to the IP3 receptor, what you have on each of these IP3 receptor subunits, you have available a inhibitory calcium binding site. So that's these orange sites here. So this is an inhibitory calcium binding site. Okay, and if calcium binds to this, it will make the chance that the IP3 receptor is going to open even lower, basically. So whenever you look at an IP3 receptor, there is some chance that it will be in the open state, okay? This is the open probability, so the chance that if you look at it, it will be in the open state. So even when there's no IP3 bound, occasionally the IP3 receptor will open. Okay, now if calcium binds to these inhibitory calcium binding sites, then the chance that the IP3 receptor will be in the open state when you look at it is even lower, basically. It's reduced even lower than it is when the calcium wasn't bound. Okay, so it's got a pretty low open probability as it is, but if calcium binds, then it will make it even lower. Okay, so you have 4 inhibitory calcium binding sites. Now, you also, on each of these IP3 receptors, have a IP3 binding site, which I'll show here in blue. Okay, so overall, the whole IP3 receptor has 4 IP3 binding sites. Okay, so these are all IP3 binding sites. Now, if IP3 goes up in the neighborhood of these IP3 receptors, then the IP3 molecules will bind to these IP3 binding sites. Okay, so you'll need 4 IP3 molecules, 1 to bind in each of these binding sites. And when the IP3 molecules bind in these binding sites, what that will trigger is a change in the conformation of the subunits of the IP3 receptor. And this change in conformation does not open the pore. I repeat, it does not open the pore. Instead, what it does is it moves this inhibitory calcium binding site so that it is no longer exposed to the cytoplasm. Okay, so let me show this. So the inhibitory calcium binding site, that's going to get covered up basically, and in its place you're going to get a new calcium binding site exposed. Okay, so here is our new calcium binding site, which I've shown now in green, and this basically is going to be an activatory calcium binding site. Okay, and now if calcium binds to this activatory calcium binding site, then that will trigger the IP3 receptor to open. So basically IP3 does not directly open the IP3 receptor. Instead, it puts it into a conformation where it can now be opened if calcium binds to the activatory calcium binding sites. Okay, now it's still a bit of a mystery where the calcium that's going to bind to these four activatory calcium binding sites, and of course you're going to need 4 calcium ions to bind, 1 in each of these activatory calcium binding sites. It's still a bit of a mystery where this calcium actually comes from, because, as I've discussed, before the IP3 receptors have opened and released calcium into the cytoplasm, calcium level in the cytoplasm is extremely low. It's around 100 nanomolar. Okay, so it's unlikely that, 4 calcium ions are concurrently all going to bind to these activatory calcium binding sites when the calcium has just come from the cytoplasm, because the calcium concentration is so low. Okay, so it's likely that you have to let in some calcium from the extracellular fluid. So calcium in the extracellular fluid is very high. Okay, so if I just draw a cell quickly here, here's the nucleus of the cell. Calcium concentration outside the cell is around 1.5 millimolar. Compare that to the 100 nanomolar that you have in the cytoplasm. So it's likely that what has to happen is you have to open some calcium channel or in the plasma membrane, which will allow a little bit of calcium to move into the cell from the extracellular fluid, and that this little bit of calcium that moves in from the extracellular fluid will then bind to these activatory calcium binding sites on the IP3 receptor. Then that will trigger the IP3 receptor to open, and calcium will then leave through the IP3 receptor from the lumen of the endoplasmic reticulum into the cytoplasm. So you will now get release of calcium into the cytoplasm of the cell. So to draw a little picture of this, okay, to draw a better picture of this basically, here's the nucleus, then we've got the endoplasmic reticulum here. What's basically going to happen, at least this is what is believed probably happens, is that some calcium comes in from the extracellular fluid. This will then activate the IP3 receptors, which have been primed by the binding of IP3, and this will allow a much bigger release of calcium from the endoplasmic reticulum into the cytoplasm of the cell. Okay, so it's actually very like calcium signaling in the heart, basically, where you allow in a bit of calcium from the extracellular fluid through the L-type voltage-gated calcium channels, and this then induces calcium-induced calcium release from the type 2 ryanodine receptors that are in the sarcoplasmic reticulum of the heart, and that then releases much more calcium from the sarcoplasmic reticulum, which is the equivalent of the endoplasmic reticulum in those cardiomyocytes. Okay, so that's how you get release of calcium from the intracellular stores. Now, what I want to talk about now is store-operated calcium entry, because calcium being released from the endoplasmic reticulum of the cell, this can trigger all sorts of downstream pathways. But when the calcium goes down in the ER, what this triggers is another pathway which I'm about to discuss, which is called store-operated calcium entry. Okay, and for short, store-operated calcium entry is abbreviated to SOCE for short. So S for store, O for operated, C for calcium, and E for entry. Okay, and what happens in store-operated calcium entry is that you bring in calcium from the extracellular fluid, which will then be moved temporarily into the cytoplasm, and then it will be moved back into the ER. Now, in this store-operated calcium entry, for a moment the calcium will be in the cytoplasm, and this calcium that's in the cytoplasm, because it's on its way to the endoplasmic reticulum and it's going through this store-operated calcium entry process, this is capable of activating all sorts of downstream pathways in the cytoplasm. And here's the amazing thing. This is what puzzled scientists and amazed scientists about this for so long. Basically, this calcium that comes in from store-operated calcium entry is capable of activating all sorts of pathways that calcium that's released from the ER, or calcium that's come in from the extracellular fluid by other means, are not capable of activating, basically. Now, why on earth should that be? Because this is just calcium. How on earth does the calcium know whether it's come in from store-operated calcium entry, or whether it's been released from the ER, or whether it's come in from the extracellular fluid? Well, basically, this involves the concept of microdomains within the cell. Okay, so this requires you to have a more grown-up understanding of what a cell actually is. Okay, so the sort of child's understanding, or at least teenager's understanding of a cell, is that, you know, it's this one great compartment of the cytoplasm. And I was guilty of this earlier on when I was explaining how the lumen was effectively equivalent to the extracellular fluid as far as these membrane-bound proteins was concerned. Okay, I said the cytoplasm is one great compartment. Well, the reality is that is not true. Instead, the cytoplasm of the cell is divided up. Imagine having partitions, basically, walls. There are walls, and you're dividing the cytoplasm up into little rooms, basically. Little places where certain things are happening, and these little compartments are called microdomains. So, basically, there will be a little microdomain, a little room, if you like, that has as its ceiling the membrane of the endoplasmic reticulum, and has as its floor the plasma membrane here, and then it's got walls separating it off from the rest of the cytoplasm. And you might ask, well, what are these walls made up of? Well, they'll be made up of the cytoskeletal proteins, such as actin being a key component. Okay, and this special little microdomain will be where store-operated calcium occurs. So the calcium that is coming in through the store-operated calcium entry pathway, which I haven't explained to you yet, but I will, is going to come into this special little microdomain, and basically calcium that's in this special microdomain will activate certain pathways that can only be activated by calcium that has come in specifically through this pathway. And the reason is that only through this pathway is calcium going to get into that microdomain, because the IP3 receptors won't be on the surface of this membrane, so you won't get release of calcium into this microdomain. Okay, you also don't have the normal calcium channels on this plasma membrane here, so you won't get calcium coming in for other reasons there. Instead, the only way that calcium can come in is through this store-operated calcium entry pathway, and then of course it's just a calcium ion, and it will activate the proteins that it's in contact with, and some of these proteins will only be in this microdomain associated with store-operated calcium entry, so they will only be activated when calcium comes in through store-operated calcium entry, and they won't be activated by calcium just being released from the ER, because that calcium won't be able to get to these proteins in here because of the walls basically separating the rest of the cytoplasm off from this microdomain. Okay, so that is basically how store-operated calcium entry is capable of activating different pathways to the calcium that's been released from the ER. Okay, so this is an important thing to understand. The IP3 pathway, the GQ pathway, does not end with the calcium being released from the ER into the cytoplasm. Instead, the story goes on, because when calcium goes down in the ER, this activates the store-operated calcium entry pathway pretty damn quickly, okay? And that then is going to bring calcium through this special microdomain here, and that calcium will then activate certain pathways in here. So not all of the pathways that will be activated by the GQ pathway are activated by calcium being released into the cytoplasm from the ER. Instead, some will be activated from calcium coming in through store-operated calcium entry. So basically we should not end the IP3 receptor pathway with calcium being released into the cytoplasm from the ER. We need to talk about store-operated calcium entry, because some pathways are only going to be activated by calcium that is coming in through store-operated calcium entry and not by the calcium that has been released from the ER. For instance, very important examples are in the activation of T cells, if you know about such things. Once the T cell has received signal 2 and signal 3, calcium activates calcineurin, okay? And if you watch my videos on the adaptive immune system, I just sort of skirt across this basically. I'm just like, signal 1, signal 2 causes calcium to go up, okay? And this activates calmodulin, and calcium calmodulin complexes bind to calcineurin. They chop off the phosphate groups off the serine residues in the serine-rich region of the nuclear factor of activated T cells, which is a transcription factor, goes into the nucleus and causes signal 3 or whatever. Okay, but that calcium that actually activates the calmodulin, which then activates the calcineurin, that is happening within one of these store-operated calcium entry microdomains. Okay, so it's actually calcium that's coming into the cell through store-operated calcium entry, and that pathway of T cell activation will only occur because of calcium coming in through store-operated calcium entry. So signal 1 and signal 2 cause depletion of calcium from the ER. So calcium does go up in the cytoplasm because of release from the ER, but it is not that calcium which actually takes the pathway of activation of the T cells further by activating the NFAT transcription factor. Instead, it's the store-operated calcium entry pathway which is activated and brings calcium into this special microdomain, which then activates the NFAT basically. Okay, so in the next video then we'll describe the store-operated calcium entry pathway, and we'll then move on to our discussion of the palmitoylation of IP3 receptors.